Hello, my name is Hannah Oliver, and the title of my research is the characterization of the Stillwater Branched1 mutants in Setaria viridis. And I would like to thank the faculty at Oklahoma State University for supporting me through my research. The Stillwater Branched1 is a gene identified in grasses such as maize, and at high levels of expression, TB1 represses lateral branching. In past studies, TB1 has shown to change the architecture of grasses, popularly in maize. And so we were curious about the effects of TB1 mutants in the model grass Setaria viridis. We hypothesized that Setaria viridis TB1 mutants would show increased branching, similar to TB1 mutants in maize. For our methods, we created two TB1 Setaria viridis mutants, which we termed as mutant 4 and mutant 5. These mutants, along with a small wild-type control group, were observed both phenotypically and genetically. For our phenotypic data, we measured domestication traits, such as time of germination and heading, tiller count at heading, height at flowering, and the primary branching time. And these were analyzed using the Wilcox and rank test. We also did a series of steps to collect our genetic data. For our genetic data, we extracted DNA from plant tissue, examined these extractions through the nanodrop program and agarose shell electrophoresis. Using DNA that we collected, we created stock solutions that ran through PCR and additional gel testing and selected sequencing. In our results, our phenotypic data showed through the Wilcox and rank test in Table 1 that between mutant populations 4 and 5, there were no significance in any of the measured traits except days of heading. And in Figure 1, when comparing the mutant populations to the wild type, we found out there was a difference in tiller count between populations. As for our genetic results, as we can see in Figure 2 and 3, we discovered two homozygous samples in the mutant 4 population. And within these two homozygous samples, we identified a 16 base pair deletion mutation that led to an altered transcript and early stop codon, which is circled in Figure 3. Our discovery results show that there were very few differences between the mutant population and among all three, with days of heading being the only significance between mutants and tiller count among all three. Hypothetically, with TB1 removed, there should appear much more lateral branching in the form of tillers than the wild type. However, because tiller count in the wild type outnumbered the mutants, as we see in Figure 1, this led us to reject our original hypothesis. And in future studies, we wish to expand our study of the domestication traits by measuring other Setaria viridis attributes, as well as discover the result of the 16 base pair deletion mutation. Additionally, we created a middle school science lesson plan following NGSS middle school life science standards. Following the 5E model of engagement, explore, explain, elaboration, evaluation, in this lesson, students learn through investigation how organism growth is influenced by both environmental and genetic factors using lima bean plants. Thank you.